Plant cells as eukaryotes are characterized by their compartmentation into membrane-enclosed reaction spaces. This structure separates different metabolic pathways, but at the same time allows a grouping of connected biochemical functions. This process allows sophisticated regulations. The borders of compartments are composed of biomembranes. These thin and highly flexible structures determine the architecture of biological systems. Biomembranes are flat asymmetrical structures, which are closed and usually topologically equivalent to the surface of a sphere or a torus. Along with the basic composition of lipids and proteins, there are a variety of individual compositions. The ratio of proteins to lipids varies within a range of 1:4 to 4:1. Both components are held together by hydrophobic interactions. The lipids form a bimolecular layer which serves as a barrier and prevents the passage of polar molecules. Integral proteins penetrate completely, or at least partly, the lipid bilayer. Conspicuous examples are the chlorophyll/B binding protein of chloroplast membranes and the adenosine triphosphate ATP synthase of mitochondrial and chloroplast membranes. Peripheral proteins bind at the membrane surface to integral proteins from which they can be detached easily by appropriate solvents. They include clathrin, spectrin, and ankyrin of the plasma membrane. Usually membrane proteins have mediating functions. They serve as receptors, ion channels at P-powered pumps, or transporters. In many cases, enzymatic activities are crucial for the function. This explains why the two sides of the membrane bilayer are usually different.